Welcome back to Simplifying Synthesis. In this video, we are going to see how Molnupiravir fights COVID. Molnupiravir was first developed at the Drug Innovation Ventures at Emory University as part of a research project into broad-spectrum antivirals aimed at influenza and equine encephalitis. It first underwent clinical trials in 2019 against influenza, and after the outbreak of COVID-19, was repurposed and studied for this disease. It showed great efficacy, with Phase 3 clinical trials reducing the risk of hospitalization or death by approximately 50%, and based on these results, was authorized for use in November of 2021. So before we can see how it works, we first need to understand the infection process of the SARS-CoV-2 virus. In the first step, the virus first enters the cell and undergoes an uncoating process which releases its proteins and genetic material. Translation of viral replication machinery then occurs, which allows for the replication of viral RNA. This RNA is then translated to produce viral structural proteins, which are assembled into a new viral particle, which is released from the cell, allowing it to affect other cells in the body, or it can be expelled, where it can go on to infect another person. In order to fight the virus, it is necessary to design a drug that can interfere with one of these steps. Drugs such as Paxlovid, which I've covered in another video, combat the virus by inhibiting the translation of viral replication machinery by inhibiting the 3CL protease. Molnupiravir, however, attacks the replication stage by inhibiting the RNA-dependent RNA polymerase, known as RdRp. So let's take a closer look at this RNA replication. RNA consists of nucleobases attached to a sugar phosphate backbone which forms long strands. New strands are formed by using the initial RNA strand as a template and attaching complementary nucleobases in a stepwise manner to build up a new strand, which can then in turn be used as another template to continue the replication cycle. There are 4 base pairs used in RNA, and they pair up in a specific manner, where a purine base will pair with a pyrimidine base. For example, adenosine always pairs with uridine, and guanosine will always pair with cytidine. We can see why this pairing occurs by looking at the chemical structure of the nucleoside. A nucleoside consists of a phosphate group forming part of the backbone in the form of a phosphodiester group. This is attached to a sugar, which in RNA is ribose, and this is bonded to a nucleobase, which in this example is guanine. When forming a base pair, the guanosine nucleotide will pair up with cytidine, which is a nucleoside which has a cytosine nucleobase. This pairing is held together by hydrogen bonds from the amine H-bond donors to the amide H-bond acceptors. A similar hydrogen bond is formed between the adenosine and uridine base pair, which again matches a purine base, which has 2 rings as part of its nucleobase, with a pyrimidine base, which only has 1 ring. So with this knowledge of the structure of the genetic material of viruses, let's look at some common strategies used in the design of antiviral drugs. One of the most common strategies is to design nucleoside analogues which terminate the chain in a growing strand of DNA or RNA. As the nucleosides are incorporated in a stepwise manner, it is possible to design molecules that mimic these natural nucleosides and can also be incorporated into the growing chain. This chain grows by binding the nucleoside at the 5 and 3 position to the phosphate backbone. In the example shown here, we can see deoxyguanosine bonded at the 5 position as a phosphodiester. This deoxyguanosine is a component of DNA, which has a deoxy sugar which lacks the hydroxyl group at the 2 position. It is the hydroxyl group at the 3 position which reacts to form the next phosphodiester and continue the growing strand. When chain-terminating antiviral drugs are incorporated into this growing strand, such as Lagevrio shown here, the strand is unable to continue growing as it lacks the hydroxyl group at the 3 position necessary to form a phosphodiester linkage. As the chain cannot grow, it halts the replication and stops the virus from reproducing and spreading. There's a problem with this strategy, however, as coronaviruses, such as SARS-CoV-2, have exonucleolytic proofreading activity that can recognize and remove misincorporated nucleotides, and can therefore repair the damage caused by these chain-terminating antiviral drugs. A solution to this problem is to use lethal mutagenesis. If we examine the structure of molnupiravir, we can see that the exocyclic nitrogen group of the pyrimidine ring can undergo a tautomerization between an N-hydroxylamine, where the carbon-nitrogen double bond lies within the ring, and an oxime, where the carbon-nitrogen double bond is found outside the ring. This tautomerization allows it to display the chemical properties of both cytidine and uridine, the two pyrimidine bases found in RNA. Molnupiravir is a prodrug which is activated in the body, first by the hydrolysis of the ester at the 5' position, revealing a primary hydroxyl group, which is then converted to the triphosphate, which is the active compound. This prodrug strategy is necessary, as they found when they administered the compound without the ester at the 5' position, it did not display the desired pharmacokinetic properties which would allow it to be distributed throughout the body. Once molnupiravir is converted into the triphosphate, it is able to be incorporated into the growing RNA strand by RdRp. Once incorporated, it can mimic the pyrimidine-nucleobases, such as in this example, where it mimics cytidine by forming a stable base pair with guanosine. This occurs in its hydroxylamine tautomer, where the hydroxylamine acts as a hydrogen bond donor and interacts with the carbonyl oxygen of the purine ring. When it's in its oxime tautomer, it is able to mimic uridine. In this case, the oxime mimics a carbonyl group and acts as a hydrogen bond acceptor by bonding to the amine of the adenosine nucleoside. As molnupiravir can mimic both of these nucleosides, it is able to carry out lethal mutagenesis. In this example, we can see a growing RNA strand with a guanosine in the templating strand, which would typically be paired with the cytidine. As molnupiravir can mimic the structure of cytidine, there is competition between these nucleobases, and it is possible for molnupiravir to be incorporated into the growing strand in place of cytidine. As this does not terminate the chain, the RNA synthesis continues as normal, except now it has molnupiravir instead of cytidine incorporated into its structure. When this newly formed strand is used as a template in a further stage of replication, there are two possibilities. One is that guanosine will be incorporated, which would be the correct pair for cytidine and would not have any adverse effect to the viral replication. Alternatively, we could have a mutagenic event, where instead of guanosine being incorporated, adenosine is incorporated in its place, as molnupiravir can bind to both bases. Incorporation of multiple bases of molnupiravir into the RNA structure leads to a buildup of mutations, and this leads to what's known as error catastrophe, where the errors in the genetic code build up and the virus is no longer able to reproduce, and thus halts the progress of the infection. So now that we know how molnupiravir works, let's look at how it's synthesized. In its original synthesis, it was made using typical carbohydrate chemistry. Starting with ribose, the hydroxyl groups are first protected as acetates, the anomeric position is converted to a chloride, and this is reacted with a sialylated uracil in a Sialyl-Hilbert-Johnson reaction. The acetate esters of the protected uridine are then hydrolyzed, and the cis-2,3-hydroxyl motif is protected as an acetonide. The ester is then introduced at the 5' position, and a triazole is incorporated into the pyrimidine ring. This can react with hydroxylamine to form the oxime, and upon deprotection, forms molnupiravir in less than a 10% overall yield in 10 total steps. While the scheme is sufficient for the pilot scale and allowed the researchers to quickly develop and test this compound, it is not ideal for large-scale synthesis due to the length of the synthesis and the cost and toxicity of the reagents used. To improve on this, Merck have developed a biocatalytic synthesis, which uses enzymes to carry out the important transformations. In this synthesis, ribose is first reacted with isobutyric anhydride using Novozyme 435 to carry out the reaction in a 99% yield. Novozyme 435 is a lipase B enzyme from Candida arctica, which is immobilized on a bead, which makes it much easier to handle. With the ester now installed, the compound was taken forward to incorporate the uracil nucleobase, again using a biocatalytic strategy. They use a 5S methylthioribose kinase to activate the 1 position by incorporating a phosphate group. This MTR enzyme was optimized through directed evolution to improve its efficiency and selectivity. The reaction also utilizes an acetate kinase, which is necessary to regenerate the ATP necessary for the reaction. With the phosphate group installed at the 1 position, they then utilized uridine phosphorylase to install the uracil nucleobase, together with the elimination of inorganic phosphate. This reacts with pyruvic acid under the action of pyruvate oxidase to produce acetyl phosphate, which goes on to form the ATP necessary for the MTR reaction, and the catalytic cycle can begin again. To install the hydroxylamine group, the compound was reacted with hydroxylamine in sulfuric acid, with hexamethyl disilazane, ammonium hydrogen sulfate, and catalytic imidazole. This was able to install the hydroxylamine group, along with the undesired silation of the 2 and 3 hydroxyl groups. This was not an issue, however, as they could be removed by simply adjusting the pH prior to the crystallization, which isolated molnupiravir in an 86% yield and a greater than 99% purity. Overall, this new method is much more efficient than the previous route, producing molnupiravir in only 3 steps with only 1 isolated intermediate, and producing the product in a high yield and purity, thanks to the novel biocatalytic cascade incorporating an innovative phosphate recycling system. Well, that's everything from this week's Simplifying Synthesis. If you enjoyed this video, please like, subscribe, and hit the notification bell. And if you have anything you'd like to see, let me know in the comments down below. As always, a big thank you to the fans who give me the motivation to keep this channel going. And an extra special thank you to all my patrons who support the channel. In the next video, I will look at the total synthesis of disifragulones A and B and disidarvarone C.